Methylated RNA Immunoprecipitation, also called MERIP, is a method designed to identify methylated groups on whole transcriptomes. It can be adapted to RNA extracts of many model organisms.
The method is simple, does not require any harsh chemical treatment, and can be used to map any RNA modification as long as specific antibodies are available.
To extract the RNA sample, first grind 200 milligrams of the plant tissue sample of interest to powder in liquid nitrogen, taking care that the tissue remains frozen throughout the procedure. Next, add 1 milliliter of RNA extraction reagent containing guanidine thiocyanate and acid phenol to the ground plant tissue, and mix well by inversion, making sure that all of the tissue is wet. After 10 minutes at room temperature, centrifuge the samples and transfer the supernatant to a new 1.5 milliliter tube. Add 200 microliters of 1-bromo-3-chloropropane to the supernatant and vigorously vortex the sample. Separate the phases by centrifugation and transfer approximately 500 microliters of the upper aqueous phase to a new 1.5 milliliter tube. Add 500 microliters of isopropanol and 50 microliters of 3 molar sodium acetate to the sample and mix well by inversion. Incubate the sample for 10 minutes at -20 degrees Celsius before precipitating the RNA by centrifugation.